Good morning, Gaza. Today in our immunology course, we're going to continue on the second lecture of the innate immunity section, and we're going to look at the following topics. The NF-kappa beta family of transcription factors. So we looked at NF-kappa beta before as a transcription factor which is able to translocate to the nucleus after pattern recognition receptors identify the presence of infection. We will also briefly look at its role in adaptive immunity as well. We'll look at another pattern recognition receptor, which is from the NOD-like receptor families, and their role in the inflammasome. So that's another cellular body which is formed in response to infection. We'd previously, in the earlier section, looked at the apoptosome. So this is today we'll look at the inflammasome. We will look at Rig-like receptors and another role for the mitochondrion in immunity. So the mitochondrion, it's not just the role at the site of the citric acid cycle in energy generation. It has a number of other roles. We looked at the role of the mitochondrion before in apoptosis, and today we'll look at its role with this class of pattern recognition receptors. I'll introduce a new pattern recognition receptor, which is a virus-induced activation of cytosolic sensors. We look at reactive oxygen species and also reactive nitrogen species and other reactive electron species, that's not just oxygen and nitrogen, which are involved in phagocytic killing of engulfed pathogens. And we look at autophagy. So it's quite a long list, but actually a small number of slides. And the idea really is to bring you to the shores of a great ocean of immunology without diving too deeply into it. So you will be paddling at the surface of or at the shore of a great ocean, and we'll just take an overview look at each of these topics. So starting with NF-kappa beta, we've already seen how NF-kappa beta is a transcription factor that can translocate to the nucleus after the destruction of the inhibitory I-kappa beta, which basically blocks a nuclear localization signal on the NF-kappa beta. And this is in response to pathogen recognition receptors such as the toll-like receptors. Now, looking at this NF-kappa beta, it's actually a dimer, as you can see actually here, it's a dimer, and it could be a homodimer or a heterodimer because there are actually 5 members of this family: NF-kappa beta 1, NF-kappa beta 2, REL-A, REL-B, and C-REL. These P65, P52, P50, P refers to basically a protein product which is sized on an SDS polyacrylamide gel by electrophoresis. So often you see proteins named simply according to the size of a band visualized by electrophoresis. So that's an experimental nomenclature, and also given a name. And sometimes there are more than one name for different gene products. And talking about the genes, it could be sometimes we can have multiple splicing variants from one gene. In this case, these are 5 distinct genes on multiple on different chromosomes, and this basically helps to explain why the same transcription factor can actually have multiple different roles. So there are many places where NF-kappa beta can be induced to translocate to the nucleus and induce gene transcription. And that's because not all NF-kappa beta is the same. So given 5 different family members and that these may be homo-heterodimers, in theory there's a huge number of possible combinations of these, and several of them have been well studied. In the next slide, we'll look at the role of NF-kappa beta actually in the adaptive immune system. What you're looking at here is the development of a B cell, a naive B cell expressing an antibody on its surface, and the development of this through various stages in the germinal center within a lymph node after exposure to antigen, and finally resulting in plasma cells which are able to secrete vast amounts of antibody. And NF-kappa beta is important in this process, just as it is in the innate inflammatory processes that we saw earlier following pathogen recognition receptor engagement by pathogen-associated molecular patterns, PAMPs. As you recall, when we looked at the anatomy of the lymph node and the germinal center, there is light zone and dark zone, and also mantle zone, and the various stages of development here, including some backward stages where we have somatic hypermutation. We'll talk a lot about that in the section on B cells and antibodies. But just the point to bring out here is that from amongst these 5 different family members, which can be paired up in various ways, we've been able to identify various areas within the B cell development in the germinal center where specific NF-kappa beta family members are important. And various experiments knocking out some of these. So we've got a REL-B double negative and NFXB2 double negative. These in italics refer to genes, and these are experiments done in knockout mice. We can see that a whole range of other genes are downregulated, while different genes are upregulated in the presence of these knockouts. So NF-kappa beta expression has a big part to play in which genes get switched on or switched off. We also have down all of these sections here represent different NF-kappa beta family members, which have been shown to be important for different parts of this process. And likewise, we can see the effect of some other family members on metabolic programming, downregulation of various metabolic programming genes, upregulation of various other metabolic genes and regulatory genes. Now, coming back to the role of NF-kappa beta in innate immunity, the NF-kappa beta, one of its targets is members of the NOD-like receptor family. And if you remember, those are NLRs, NOD-like receptor families. And these respond to DAMPs and PAMPs. The unusual thing about these are that they are cytosolic. They're not bound to, or not initially bound to membranes. And they have a 2-stage activation. So first of all, other pathogen recognition receptors, such as toll-like receptors, are engaged by pathogen-associated molecular patterns, and then we have that NF-kappa beta signaling pathway, which we've looked at before. Thereby, we get upregulation of these NOD-like receptors. It's a big family. The most well-studied of these is called NLRP3. And these basically exist in a monomeric form, and they can also form a large complex structure in association with caspase 1. And this structure, which is indicated in front of you here, is called the inflammasome. So it's a distinct body within the cell which is involved in inflammation. When we talk about engagement of pathogen recognition receptors, what we're looking at is an inflammatory process mediated by a series of cell signaling events in response to the presence of pathogen. There's still a huge amount of research ongoing to try to understand better the physiological role of these. Their role in a huge number of autoimmune-related diseases is becoming well established, and these are important targets for therapeutic intervention or for potential therapeutic interventions. The NOD-like receptor family have several subfamilies. The most well studied is NLRP, and shown here is the the domain structure of various NLRP proteins. The most important one that I will just briefly mention is this domain here, the Nacht domain, and this facilitates the self-oligomerization. In order to form the inflammasome, this protein changes from being in a monomeric form to being in that pretty star-type shape that you saw in the previous slide. After the expression of NLRP following NF-kappa beta translocation to the nucleus, in order for this protein to function or begin its function as the inflammasome, then there needs to be a series of post-translational modifications, and we can look above and below to look at positive and negative regulators of the activity of NLRP3. Right on the top here I listed a whole series of enzymes, and these enzymes function in order to direct two types of or three types of activity. One is phosphorylation, and here for example is serine 198. In addition, we have the dephosphorylation, so tyrosine, Y stands for tyrosine. This can be phosphorylated. If it's dephosphorylated by this enzyme, then it becomes active. We have dephosphorylation sites here, and the U here stands for ubiquitin, and some enzymes facilitate the ubiquitinylation, meaning the adding of a protein called ubiquitin to at this position. In addition, we have enzymes which remove ubiquitin. The action of all of these enzymes resulting in either the addition or removal of a phosphate group or a ubiquitin group, those act to direct the NLRP3 to form the inflammasome. In addition, we have negative regulatory controls, again a whole series of enzymes, also which direct changes in phosphorylation status. So several phosphorylation activities by this enzyme at various different tyrosine residues, again tyrosine here, serine, and ubiquitin. The question marks are because the actual site of ubiquitinylation is not clear. Here it's clear. K stands for lysine, and lysine is a residue which is targeted for ubiquitinylation, and that's at position 495. In addition to the post-translational modifications on the previous slide, something else needs to happen. So here we have, first of all, going backwards a little bit, starting from the pathogen recognition receptor, TLR4, we have a process whereby we will end up with the production of NLRP3, but NLRP3 is not active initially. We need those post-translational modifications. In addition, there's a second effect which needs to take place before we get to the active NLRP3, which can form itself into the active NLRP3 inflammasome in association with other proteins, including an important effector protein called caspase 1. And that other stage of activation is not very well understood, but a series of changes that can take place in the cell under various inflammatory conditions, including changes to the mitochondria, and that's what's shown in this slide. Also changes in the trans-Golgi network, actually in response to infection and inflammation, result in this this final switching to the active NLRP3 inflammasome. There are a lot of things on this slide which it's too advanced for, or it's not necessary to go into all of it for undergraduate level, but just to take in mind the concept that now, following these two stages of activation, first of all, the production of NLRP3 and its post-translational modification in response to initial detection of some pathogen-associated molecular pattern, followed by some general changes that take place in an infected cell, which are somehow sensed and the mechanism of that isn't really understood, as indicated by these dotted arrows. We don't really know how these changes are actually causing NLRP3 to go into forming the inflammasome, but these changes take place. And perhaps one of the most important, or at least the most well studied of these results or effects of this, is that caspase 1 becomes active within the inflammasome and is able to cleave some interleukins from a pro form into an active form. And these interleukins then change the cellular environment, causing paracrine signaling, resulting in further downstream effects in the innate immune system. Though we have many bits and pieces of the molecular mechanisms or molecular events, still we have many gaps in understanding. Nevertheless, all of this huge list of autoimmune diseases here are all associated in some way with dysregulation of NOD-like receptors. Autoimmune thyroiditis, type 1 diabetes, vitiligo, inflammatory bowel disease, arthritis, psoriasis, systemic lupus erythematosus, autoimmune hepatitis, celiac disease, multiple sclerosis. Huge number of autoimmune diseases where misregulation of this very important cellular inflammatory process results in disease. People are looking at trying to target the NLRP3 inflammasome in order to be able to counteract these diseases. The tricky thing is that the NLRP3 inflammasome is part of our normal defense against infection and also against tumorigenic cells. So it's a difficult area, and it's all an issue of balance. Now we'll move on to another pathogen recognition receptor, and this is RIG1 and RIG1-like receptors. Here are some examples of these receptors that have been more well studied amongst the wider family. These indicate the various domain structures of the proteins, and the take-home message mainly from this slide, these are cytosolic pathogen recognition receptors like the NOD-like receptors. The interesting thing about these ones is that maybe similarly in some way to what we saw for NLRP3, that the active form is a association between the receptor and other proteins. In this case, there's an adapter protein which needs to interact with this one through these so-called card domains, which are present in the adapter and the actual receptors themselves, the RLRs, which are up here. These are called MAB. These adapters are called MABs, which stands for mitochondrial antiviral signaling. And this is a protein which exists in the outer membrane of the mitochondria. And it's the association between this and this is responsible for the downstream effects of the RIG1 and RIG1-like receptors. Here is a mitochondria, and here we have these adapter proteins sitting on here, the MABs, and here is the RIG1 as a prototype of the RIG1-like receptors, and it's folded in such a form as to be unable to interact with the MABs. These blue things here are the card domains, which in the case of RIG1 are not free to interact with the MABs sitting on the surface of the mitochondria. However, RNA viruses can be detected by this receptor, and the detection of and the binding of viral RNA, double-stranded viral RNA, results in a conformational change which releases the card1 domains, enabling them to be interacting with these adapter MABs on the surface of the mitochondria. So again, another interesting role for the mitochondria. The mitochondria does so many things apart from what you studied in maybe in more introductory courses in terms of its role in metabolism. Now coming to something, at least something which is familiar. I know it sounds the whole thing seems complicated to you. Engagement of these pathogen recognition receptors, translocation to the surface of the mitochondria and forming these clusters here, results in recruitment of various other molecules, some of which are shown here, which lead to the translocation of NF-kappa beta. So this part is, as we saw before, that a kinase here is able to remove the inhibitory molecule, which isn't shown here, but it's just the same concept as in the earlier slide where I showed you NF-kappa beta. It's removal of the inhibitory molecule by this kinase, which phosphorylates the inhibitor and leads to its destruction in the proteasome, enables the release of NF-kappa beta, which has a nuclear localization signal. It's the same story as before, which can translocate to the nucleus, therefore, and activate gene expression. Remember, this is a family, so depending upon which family members are forming this dimer will determine exactly which set of genes get transcribed. In addition, we have the activation of IRF3 and IRF7, and these are this stands for IRF stands for I've forgotten that, not interleukin, no, interferon. Sorry, I don't know why it slipped my mind. It's interferon regulatory factor. So remember we looked earlier at the interferon response. So these are responsible for that. NF-kappa beta is also part of this story. Okay, now we just take a little break away from our subject. Perhaps this slide is a little bit out of place, but we could have had this in the earlier discussion in the previous section where we looked at the different types of innate immune cells and have dendritic cells and macrophages. The odd thing is that monocytes are able to be converted into dendritic cells or macrophages. And one of the interesting important things about this is that we can control this, and we can actually use innate immune cells as a therapy. So here's an example of controlling the differentiation of monocytes after their leukapheresis, separating out monocytes from the blood, and these can be cultured, and we can control their differentiation depending on which factors, growth factors and interleukins we add. So if we understand all these cytokine signaling, then we can use that knowledge to our advantage in cellular engineering, and we can control the differentiation of monocytes into dendritic cells, and we could use these. And here's an example of a paper with some technical developments seeking to make a novel type of vaccine for colorectal cancer. There's a big area of study how to develop vaccines for cancers, and we'll look at this again as later from some other aspects. But from the innate immune cell, then if we can take what we can call neoepitopes, the word's not on the slide, but these are epitopes specific for this type of colorectal cancer, and we can mix these with the dendritic cells, load them up, and then we can add these back to the patient. We've basically made now a weapon which is going to find its way into lymph nodes and present those antigens that we've given it. It's going to present those to T cells and therefore stimulate the adaptive immune response. So that's an area of a novel type of vaccine therapy that people are interested in, which probably you may not have come across before. And another cytosolic pathogen receptor is a cytosolic DNA sensor called STING, which stands for Stimulator of Interferon Genes. Again, these interferon genes are important. There's a lot happening here, but let's just focus in, or let's start with some of the simple things. Here we have gene expression, our old friend NF-kappa beta, and our old friend interferon regulatory factors. These are going to translocate to the nucleus, so that's finally what's going to happen. The receptor here we have it sting. It's on the endoplasmic reticulum, and it's able to recognize an unusual molecule called cyclic GAMP, which is produced by an enzyme in response to a double-stranded DNA sitting in the cytosol, which shouldn't be there. All the DNA should be here or in the mitochondria. We have some DNA in the mitochondria. The DNA floating around in the cytosol is indicative of a viral infection from a DNA virus, such as a pox virus. And this is recognized by C-gas, and that recognition results in the formation from ATP and GTP of cyclic GAMP, which is recognized by this intracellular DNA sensor. So it's indirectly, this is black thing is indirectly recognizing the presence of double-stranded DNA, finally leading to the production of various interferons and other antiviral genes. And here's just an example of some different types of viruses: HIV, herpes simplex virus 1, and different ways in which that sting here, sitting on the endoplasmic reticulum, is able to be activated. And I don't want to go into the little molecular details here, but DNA can fold up into certain types of structures which can be recognized. So in this example, it's single-stranded DNA which folds up on itself. Also, RNA-DNA complexes can also be recognized. Strangely, some viruses actually contain within their virion that cyclic GAMP, which therefore is able to be recognized directly by sting. I'm sure the virus is not, it doesn't have an objective to have this in its virion, but in the process of formation of the viral capsid, then it seems that somehow cells are able to ensure the incorporation of this, which is beneficial to the cell. Now we'll move on from this to just have a quick look at phagocytes, and those are important innate immune cells that actually directly go and seek out pathogens and go and kill them. And so here you have, for example, a neutrophil. Neutrophils can digest the whole bacterium in a very, very short period of time, and this is a scanning electron micrograph of a neutrophil phagocytosing anthrax, a particularly nasty bacterium, has been experimented with for use in biological warfare. Various types of killing agents within granules or within lysosomes. We have ROS and RNS. ROS stands for reactive oxygen species, and this one is a reactive nitrogen species. Also, some proteins are present within these which have some killing activity. Reactive oxygen species, reactive nitrogen species. Hydrogen peroxide can be can activate chlorine in order to make hypochlorous acid. All of these are very nasty, destructive molecules for bacteria, and the granules are full of these things. In addition, here perhaps a little bit complex, singlet oxygen. You probably haven't heard of that. It's a quantum state of oxygen, which is a little bit different to the normal state of molecular oxygen. And this one up here just indicates this unusual quantum state, which is basically it's very active. The little dot here represents a free, an unpaired electron. So the at the chemical level, it's a little bit complex, but there are different ways in which some molecules, so oxygen or nitrogen, can be much more reactive than normal. And there's some physical chemistry here, which if you're interested in, then here are the details. But I'm not going to ask you any questions about it in any of the assignments. But I was interested just to show it to you because Nature is quite a prestigious journal, and there's an interesting article here showing that this unusual quantum state of oxygen actually has a medical effect in terms of controlling vascular tone and blood pressure during inflammation. As you can imagine, it's not an easy thing to study within a biological organism, but it's something that you may hear more about later. Perhaps there's nothing new. It's just another slide. Here we have a phagocytic cell, and just trying to indicate to you here various enzymes which are basically producing these reactive oxygen and reactive nitrogen species, which after phagocytosis of pathogens are actively going about leading to their destruction. And finally, in today's lecture, I've just mentioned to you autophagy. So phagocytosis, I think you can have a, sorry about that noise, you can have a fair picture of what phagocytosis is. What about autophagy? Autophagy comes in many different forms, as indicated by all these colored segments here. We're interested in this one here called xenophagy. Xeno means something alien or something foreign. Phagy, like phagocytosis. The difference between xenophagy as a type of autophagy and phagocytosis is that the pathogen is already inside the cell. So it's not the case of, say, a neutrophil which is seeking out an anthrax bacilli and grabbing hold of it and endocytosing it and then releasing all these nasty reactive oxygen species and reactive nitrogen species to destroy it. Rather, in the case of xenophagy, a pathogen has already got inside the cell, and the cell is able to detect the presence of this and basically build a membrane around that pathogen. And these are various important receptors and proteins. Our friend ubiquitin is there again. This UB stands for ubiquitin, that you saw when I discussed the NLRP3 post-translational modification. Ubiquitin crops up in many cellular processes. Ubiquitin, the most common place where students hear about ubiquitin is as a tag attached onto proteins to direct them to the proteasome for destruction. Earlier in today's lecture, you looked at ubiquitin as a regulatory molecule on the NLRP3 in order to make it able to form the active inflammasome. And now we have another role of ubiquitin here. So regardless of all these molecular details, the idea being that once you have a pathogen which has got inside a cell, then there is a mechanism in place to form a phagosome around that, and then that is directed towards the lysosome. So the lysosome, definitely in cell biology, you came across that before, and that's where lysis takes place. So anything that's directed towards the lysosome is basically being directed towards its destruction. So it's another way of clearing pathogens. As we talked about mitochondria before, we have a version of autophagy called mitophagy, which is basically getting rid of old mitochondria. And I'm not going to talk about all these other types of autophagy. Okay, it's enough for us, the xenophagy. Right, so this will conclude today's lecture. And the next lecture we'll finish, I plan to finish the topic of innate immunity. Innate immunity is perhaps the one which you'll find most complex. In the next lecture, we'll finish off the subject with the role of the complement system, and then we'll move on and we'll look at B cells and T cells. Okay. Look forward to seeing you next time. Very soon now I'm going to make another assignment, and basically I use the assignments as a kind of a self-learning tool. So the assignments will be on the material we've covered up so far in this innate immune section, and it's a self-learning tool based on the difficulties most of you have, that some of you can't even get to see, hardly see this video because of the state of the internet under your bad situation in Gaza. So this will lead you through the slides which you already have, and they're fairly straightforward questions, but if you're following the slides, the questions will actually be part of the explanation process. So if anything wasn't clear in the slides, the questions will direct you towards that, which will let you see the main concept. So it's all these little details that you see on the slides. I'll only be picking up those things which highlight overall general concepts. So in addition to being part of your major or your only evaluation, this is also a learning process at the same time. Okay, so we'll call it a day until we meet up for the next lecture. Assalamu alaikum.